guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about impact of gene mutation on human health. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So, gene mutation represents mutation in the single gene. It causes different disorders which are transmitted to the offspring. Most common genetic disorders are color blindness, hemophilia, sickle cell anemia, phenylketonuria, thalassemia. So we will talk about them one by one. First is color blindness. Color blindness is a X-linked recessive disorder due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. So actually our eye contains three types of cone cells: blue, green and red. In color blindness, red and green cones are defective, and these persons fail to discriminate between red and green color. It occurs in 8% of males and 0.4% of females, since it is a X-linked recessive disorder. Next is hemophilia. It is a X-linked recessive disorder. In this disease, a single protein that is involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. So we know that blood clotting mechanism requires many proteins, which are called blood clotting factors. If one of them is affected, the whole blood clotting mechanism gets affected. So it happens in hemophilia. One of the blood clotting factors is affected, which causes non-stop bleeding even after a small cut. Next is sickle cell anemia. So this is an autosome linked recessive trait. The defect is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. So in my previous lecture, I have explained about this disease in detail. Here I am just giving you the overview. Hemoglobin is made up of two types of protein chains, alpha and beta. In sickle cell anemia, beta chain is affected. The 6th amino acid of a normal beta protein is glutamic acid. When the mutation occurs, this glutamic acid gets substituted by valine. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. So this is the cause of the substitution. The codon is GAG, which codes for glutamic acid normally. A substitution mutation occurs and changes the codon GAG to GUG, which codes for valine. The mutant hemoglobin molecule undergoes polymerization, causing the change in the shape of the red blood cell from biconcave disc to elongated sickle-like structure. So the faulty hemoglobin molecules accumulate in red blood cells and change the shape of the cell. It causes deficiency of healthy red blood cells and severe anemia. Next is phenylketonuria. This is an autosomal recessive trait. It causes error in metabolism. the affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of these, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. So, phenylalanine can't be converted into tyrosine. As a result, phenylalanine is converted in some other ways. It yields some unusual toxic chemicals such as phenylpyruvic acid, etc., which accumulate in brain and cause mental retardation. So, accumulation of these in brain results in mental retardation, and these are also excreted through urine because of its poor absorption by kidney. And these chemicals are not efficiently absorbed by kidney, hence excreted through urine. Next is thalassemia. This is an autosomal recessive trait. Thalassemia is of two types, alpha thalassemia and beta thalassemia. In alpha thalassemia, production of alpha globin protein of hemoglobin is affected. In beta thalassemia, production of beta globin protein of hemoglobin is affected. Both cause the abnormal hemoglobin molecules resulting in anemia. Alpha thalassemia is controlled by two genes HbA1 and HbA2 on chromosome 16. Hb is because of hemoglobin and A is due to the alpha. A mutation may occur in one or more of the four genes. The more genes affected, the less alpha globin molecules produced. Here, 4 genes mean 2 alleles of HbA1 and 2 alleles of HbA2. So, mutation may occur in one allele or 2, 3 or all 4. Depending on these, severity of the disease results in. If one allele is affected, some amounts of alpha chain get produced. But if 2, 3 or 4 genes are affected, production of the protein gets reduced accordingly. So we can say that thalassemia is not a qualitative, rather a quantitative problem of synthesizing globin molecule. Beta thalassemia is controlled by a single gene HBB on chromosome 11. HB codes for hemoglobin and B is due to beta. So this is all about today's lecture. I hope you liked the lecture. If you want to get the PDF notes of this topic, please find the description box or first pinned comment. Thank you.